Yes, so far what we have established in the chapter is the anatomy, the histology and the physiology. Last time we did the physiology of urine formation, that is 3 steps: ultrafiltration, selective reabsorption and tubular secretion. Right? Now for today we need to have a look at 2 things. First, the filtrate which is formed, how is it concentrated? That is number 1. And second, how is the regulation of this kidney function achieved? Right? Every system has a regulator. Right? In case things go a little up and down, you need to regulate these processes. So what is the process of regulation of the kidney function? Now the entire filtration part, right, the first ultrafiltration, is achieved by means of the cortex area, that is Bowman's capsule and glomerulus. The concentration part is achieved by the medulla, as you're talking about the loop of Henle, right? So if we can just draw, so this is the cortex, the medulla. So both the limbs of the loop of Henle are lying in the medulla. So let's draw that. So is this the descending limb of loop of Henle? And this should be the ascending limb of the loop of Henle. In case of the juxtamedullary nephron, we have 2 types of nephrons, cortical and juxtamedullary. Do we have a vessel surrounding the loop of Henle which is called as the vasa recta? So let's draw a vasa recta here. The vasa recta. So would this be the descending limb of vasa recta, and this would be the ascending limb of the vasa recta, right? This is the place where the concentration of fluid is going to occur. So obviously now concentration becomes the purview of which type of nephrons? Juxtamedullary nephrons. Great. Now all these structures are lying inside the medulla, or can I say the medullary tissue, right? And 1 million such structures are there. They are not spaced out like this, huh? Just for the diagram we have made it spaced out, please. They are lying very close to each other. Now this medullary tissue, we will call it as medullary interstitium. So this white area here is the medullary interstitium. Now this interstitium has the constant presence of 2 substances. NaCl and urea. Now see, when you have a tissue, to maintain the tissue in a living state, you require it to be surrounded by fluid. Tissue dry ho jayega to mar jayega, right? So there is a fluid inside the interstitium, so which we'll call it a medullary interstitial fluid. So what is the fluid called as? Medullary interstitial fluid. And in the fluid we have got 2 main characters. One is NaCl, one is urea. So are NaCl urea solute molecules present in a liquid now? So the number of these molecules in a liter of solution is its osmolarity. So how much NaCl and urea solute molecules are present in a liter of solution is defining its osmolarity. So one more term comes into picture here now, osmolarity. How do you define osmolarity? The number of solute molecules in a liter of solution. And the unit of osmolarity is osmoles per liter. Osmoles per liter. How do you have a kilogram for weight? Right ना? अगर किलोग्राम से कम होगा तो ग्राम्स, ग्राम से कम होगा तो मिलीग्राम्स. So वैसे ही osmoles से कम रहा तो milliosmoles, a minute division of the weight now. Now when you look at the osmolarity in the interstitium, when you look at the osmolarity in the interstitium, is interstitium